We're going to walk through the brain today. So we'll do like the anatomy of the brain, different parts and what they do. Honestly, y'all, we could spend an entire semester just on nervous tissue and the brain and the spinal cord and the nervous system in general. And so we are definitely not covering every anatomical area in the brain. We're not covering all the structural and functional areas of the brain. We're just picking out a few of them, okay? And then we're going to go through the protection. Obviously, nervous tissue is very soft. The way I think of it, if any of you if you've ever hunted or your, parents have hunted, if you have ever like field dressed a deer, for example, you know that fresh organs feel very different from preserved organs. And if you've ever come in contact with fresh nervous tissue, it is almost like Jell-O. It is so, so soft. So we have to have ways that we can really protect that tissue. So we're going to walk through that. And then we'll talk just about a few disorders. We'll talk about things like meningitis, for example, that affects some of the layers. We call them meninges that cover the brain. And then on Wednesday, like I said, we'll get into the spinal cord. Okay, so today is all on the brain. Now, what I'll do in the lecture today is I'll kind of pop back and forth between a whiteboard and the PowerPoint. And really all I'm doing on the whiteboard is I'm just going to kind of keep an outline going. So you can write this down, jot it down as I jot it down on a piece of paper. I think it's really helpful so that you can sort of see how these different parts of the brain fit together and what they're composed of. Okay, so these are the 4 areas of the brain that we're going to talk about today. So we're going to do, we'll talk about the cerebrum and parts of the cerebrum. We'll talk about the diencephalon, the brainstem, and the cerebellum. So I'm going to pop over to this whiteboard. Okay, and I'm just going to write these on the whiteboard so we can keep a running tally. So we're going to start with the cerebrum. And then we're going to do the diencephalon. And the brainstem. And I got to move some tools out of the way. And the cerebellum. Okay, so those are the parts of the brain that we're going to walk through. Okay, the 4 parts. It is so hard for me to write on this whiteboard. I swear my handwriting is way better than this. All right, so let's pop back over to our PowerPoint. All right, so these are all the parts we're going to talk about. So for the cerebrum, this is where we're starting. The cerebrum, is basically, if you think about the brain, it's all the big part of the brain you're looking at. And the cerebrum, when you look at it, if you look at a brain, it's not nice and smooth. Instead, it has all these ridges and grooves in it. So I'm not gonna be able to write on here. So the cerebrum, this part right here, it has these elevated ridges, which hopefully you can still see my mouse. These elevated ridges are here. So you can see this big ridge right here. And then you can see these grooves, these sulci, these kind of dip down. So we have gyri and sulci. So gyri is actually plural. The singular form of gyri is gyrus. So instead of an I at the end, it's US. I would write that if I could. So that's gyrus. And that would just be 1. So 1 ridge is a gyrus. Multiple ridges are gyri. Oh, here you can see it right here, gyrus. And then same thing for sulci. A sulcus is just 1 groove. Many of those grooves dipping down are sulci, okay? Now, a fissure is a deeper groove. So a sulcus is a shallow groove. So you can see over here in this cross section, you can see a sulcus. And then next to it, you can see a fissure. It's just deeper. And so some examples of fissure. Fissures would be like right here. This is called the transverse fissure. This is a really deep groove, and it separates basically the cerebrum, so this big part up in here, from the cerebellum, this part down here. Okay, so that is a fissure. We also have another fissure, which you can't see from this angle. It is a fissure that runs right down the middle of the brain. We call that the longitudinal fissure, and it separates the right and left hemispheres of your cerebrum. Okay, so a fissure is just really deep. A sulcus is just more shallow. Okay, so all these little grooves that you can see in here, those are all sulci. All right, so the cerebrum, it has three basic regions that I'm going to walk you through. The cortex is on the outside. It's the part you can see. So let's go back. All of this in here, this is all the cortex, okay? And the cortex is gray matter on the outside. Now, we talked a little bit in the last chapter about what makes the difference between white matter and gray matter. White matter is white because there's a lot of myelin. Remember, myelin is that fatty covering that we find on our axons. It insulates them and it sends impulses faster that way. So the reason why white matter is white is because you have a lot of myelinated axons. The reason gray matter is gray is because you don't have a lot of myelin. So the cortex is gray matter. So on the very outside is gray matter. It's just darker in color. I don't know if I have a picture, maybe, I guess sort of here. They've colored the gray matter red and blue here. So you can see it's just on the very outside. Okay. The white matter is just underneath that. So if you were to like take a probe and stick it through the cerebrum, you'd go through the gray matter into some white matter. And again, white matter is white because of all of the myelinated axons. And that means that in your white matter, really its main job is to send signals very quickly. Communication. When I say communication, I don't mean you communicating. I mean neurons communicating very quickly. And then deeper to that are these little pockets of gray matter called basal nuclei. Okay, so those are the three parts of the cerebrum that we're going to walk through. And we're going to start with the cortex. So your cortex is this superficial gray matter. Again, let's see if I can kind of get close to where I'm supposed to write it in. Not even remotely. Let's try it this way. Maybe this will work. Oh, there we go. We'll do it this way. All right, so the superficial gray matter, sorry. This makes up about 40% of the mass of your brain. So this outside part with gray matter is 40% of the mass of your brain. That's huge. And really what it's doing is lots of different functions. So this outside part of your cortex is there because it allows you to pick up sensations. Again, think of your five senses: touch, taste, sound, sight, smell. That's your cortex. It's picking up all of that information. So it is there for sensations. It is there for communication. Literally you communicating, you speaking, but also communicating between different areas of the cortex. It's also very helpful in memory, so storing memories and understanding. And then also it plays a huge role in voluntary movements. Okay, so we're going to go through all of these, right? We're going to talk a lot about sensation and movements as we go through this cerebral cortex. Now, another thing I want to point out is that your cortex, each hemisphere, so each side, you have a right side and a left side, each side acts contralaterally, meaning the right side of your cortex controls the left side of your body. The left side of your cortex controls the right side of your body, okay? So they act contralaterally, and your hemispheres are not equal in their function. And you're going to see this as we talk about the cortex. You have the speech area, which helps you form speech and move all the muscles involved in creating speech and sound, only in one hemisphere. You don't have it in the other. So they're definitely not equal in their function. But also your hemispheres are not equal in their function in that you have one hemisphere that's typically dominant. It's doing the majority of the work. The other hemisphere helps, but it's not the dominant hemisphere. Okay, so you may have heard of somebody being referred to as right-brained or left-brained. So we're going to talk about that as well. And then also, none of these functional areas in the cortex, the sensory area, the motor area, the communication area, the memory area, none of them ever work alone. They're always working together. And so we'll also talk about that. And that is a pretty important concept, is that these areas are all working together. Okay. Oh, darn it. I knew that was going to happen. So sometimes when you add these tools on here, when I have to use this, it is going to keep all my drawings from slide to slide. Yeah, that's still way off, unfortunately. All right. So there's 3 of the functional areas that we're going to walk through today. 3 main functional areas. So there are areas that control your voluntary movements. So these are what we call motor areas. Now I'm going to see, I'm going to draw these in red, write motor area in red, because motor areas in your textbook are always colored in red, okay? So the motor areas, these are controlling voluntary movements. These are typically in, darn it. These are typically, when we look at the brain, the motor areas are typically in the front of the cortex. So I'll just write front. These are anterior. And again, they're usually red or pink in color. Now we also have sensory areas, and I'm going to write these in blue because they're blue in your book. So your book takes the cortex and colors in blue these sensory areas. These are involved in sensation, conscious awareness of sensation. Again, think of your 5 senses: touch, taste, sound, sight, and smell. And so these are always typically located in the back of the brain. Okay, so when I say front and back, we actually have a fissure that separates the front half from the back half of your brain. So motor areas are in the front, sensory areas are typically located towards the back. And then we also have, and I'm going to keep these in blue, we also have what are called association areas. And association areas, I colored them in blue because a lot of times they are linked with our sensory areas. They help you interpret the sensation, right? So maybe like smell, for example. Have you ever heard that sense of smell and what you smell really can bring back memories? So like, you know, my grandma passed away when I was in high school, but she wore this perfume, and anytime I smell somebody that has that on, I instantly think of my grandma, right? And so the smell and the memory of that, those are highly linked together. So the smell is the sensory area, but the memory would be the association area, right? So we typically link association and sensory areas together. All right, so this is an image that is just showing you some of the functional areas of the cortex, right? So again, you know, I mentioned to you that we can see that the front half is kind of colored in this pink or red color. The back half of the brain is in blue, right? We're looking at it from a side. And so I told you that we have this big groove that separates the front and the back half, and we call that the central sulcus. So the central sulcus, it's not a fissure, it is a sulcus, so it is not super deep, but it separates the front half from the back half, okay? The motor half from the sensory half in the back. So we're going to walk through our motor areas first, and then we'll go through the sensory areas. So the motor areas, these always lie in front of the central sulcus. So I said front before. I probably should have said anterior instead of front. So these are all, again, here's our central sulcus. So these are lying in front of the central sulcus. And so we have 4 main motor areas I'm going to walk you through. We have more than this. I am just walking you through 4. The one that is here in this sort of bright red color, I'll circle it. That is called the primary motor cortex. Okay, so this is involved in voluntary motor movement. Let's pick another color here. All right, so this one right here that I'm drawing in sort of a maroon color, I'm circling that one, sits right in front of the primary motor cortex. This is called the premotor cortex. Okay, this one is more involved in learned repetitious patterned behavior. So like playing the piano or an instrument or typing on a keyboard. That's going to be more from the premotor cortex. We also have, I'll do this one in another color. We also have this area right here called the frontal eye field. This one obviously is going to control voluntary eye movements. And then I mentioned to you earlier that we have this area that is involved in speech. It is this one with the dashed line around it. And that's called the Broca's area. This one is heavily involved in the movement of all the muscles required for you to speak. This is actually only found in the left hemisphere. It's not found in the right hemisphere. Okay, so what we're going to do is go over each of these motor areas. So we'll do all 4 of these areas. So we're going to start with this primary motor cortex. Okay, so the primary motor cortex is this one right here. It's this bright red one. And we say that it's found in the precentral gyrus. Okay, so let's think about that word. Precentral gyrus. Pre means before. Okay, so remember we have the central sulcus right here, this groove. Remember a gyrus is sort of an elevated ridge. So precentral means it's the gyrus, this ridge, before the central sulcus, the precentral gyrus. And you're going to have a lot of neurons in this primary motor cortex that have really long axons. And the axons go all the way down into the spinal cord, and they make up what's called the corticospinal tracts. Cortico means cortex, and spinal is spinal cord. So these are really long axons that are basically running from the cortex all the way down to the spinal cord. And again, the function of primary motor cortex is conscious control of voluntary movements. So being able to move around, have conscious control over that. All right, so this is an image. I actually think this is super fascinating. So this image right here, this is, we are actually only, for our purposes right now, we are only going to look at the left side right here, which is colored in red. We're talking about motor. So this is looking at the primary motor cortex, and it's looking at the different areas of the cortex and what areas of your body they control. So there's been a lot of study on obviously the brain and how it works. And so I think this is super fascinating. So they have been able to identify exactly what area of your motor cortex controls your hand. What area controls your leg? What area is controlling your face and the muscles of your face? And so you can see here on the left, this is the primary motor cortex in red, and you can see that they've sort of superimposed parts of the body all over this, right? And so we actually call this drawing the homunculus. And the term homunculus means little man. Right? And it kind of makes sense. It's like a little man that's sort of superimposed here. And so one of the things that you're going to notice when you look at this drawing here with all these parts and pieces, first you're going to notice it's definitely not in order. It's not head to toe, right? You can see like You know, here's the tongue and then the face, and there's this huge hand and a trunk and a leg. So it's not like, it doesn't look like a man per se. So you'll notice it's out of order. And the other thing that you're going to notice is that some of the areas on the body are drawn really big. Like the face is huge and the hand is really big. So let's talk about both of those. So the reason why it's out of order, obviously, is because they drew the part of the body that that particular area controls. So it can't be in order. It can't look like a, you know, a stick figure per se. And then the reason why you have some areas that are larger than others is because the larger it is, the more fine motor control you have. Meaning you have much more fine motor control in that area. So like the face, you have very fine control of the muscles of the face, right? To provide different facial expressions. You have very fine control over the muscles that are in your hands and your fingers. You do not have fine control over large weight-bearing muscles, like muscles in the legs and the thighs and the trunk. Those are big weight-bearing muscles. You're not going to have fine motor control over those, right? So they're drawn larger if they have a lot of fine motor control. Okay. So that is the primary motor cortex. Now let's look at this area right in front of it. So this area right in here, it's sort of this pink area. Let me annotate on top. This area right here. This is called the premotor cortex. So this sits right in front of the precentral gyrus. Okay, so it's the one right in front of the primary motor cortex. And the premotor cortex is involved in learned, repetitious, patterned motor responses. So again, this would be like playing an instrument or being able to type on a keyboard. Those are all learned, repetitious, patterned behaviors. So this one's really helpful in coordinating sequential actions. Now, Broca's area is this area right here with this dashed line around it. Again, this is only present in one hemisphere. It's usually present in the left hemisphere. And so again, remember at the beginning of the lecture, I told you that your hemispheres are not equal in their function. Usually one hemisphere is dominant over another. So the Broca's area is there as a motor speech area. It directs all the muscles of the face and the tongue and helps you form words. So that makes sense, right? We are talking right now in all these red colored areas, we're talking about motor, movement, right? We've got this area right here, primary motor cortex. It's involved in movement. Premotor cortex, it's involved in movement, right? Broca's area is involved in movement. One thing though that I think is pretty fascinating about Broca's area, they have found that this area is not just involved in movement and muscle movement to get you to speak. This one is activated when you are even thinking about what you're going to say. So let's say you're in class and you raise your hand and you are thinking about how you're going to ask that question. Broca's area is activated. So you may not be even speaking yet, but Broca's area is activated. So I think that is really fascinating. So it is active even as you're preparing to speak, which is interesting for a motor area. And then the last area that I'll mention, another motor area, is this one right here. This is our frontal eye field. And the frontal eye field just controls voluntary eye movements. So it allows you to look around, just eye movements. So now we're going to move back behind that central sulcus and we're going to look at our sensory areas that are in the back. Now remember, the sensory areas that are in the back of the brain, we call that the posterior area. These are all going to be colored in blue. And these are all involved in the awareness of your 5 senses: touch, taste, sound, sight, and smell. Okay. So we say that these are going to lie posterior to the central sulcus. And the sensory areas that we're going to walk through back here, we have a primary somatosensory cortex. This is basically involved in touch. Now, remember when we started this, I told you that we also have these areas called association areas. They help us sort of store memories of these sensations. And so for all of these areas, they're all going to have an association area with them. So the primary somatosensory cortex picks up touch, and it also has an association cortex. It helps you interpret touch. So for example, if somebody comes up behind you and they tap you on the right shoulder, you know they tapped you on the right shoulder, not on your ear or on your leg. You know exactly where it was, okay? That's because of the association area. We have visual areas. Obviously, those are going to pick up sight. Auditory areas, which are picking up sound. Olfactory areas, these will pick up, not taste. Hang on, let me erase. These will pick up smell. Y'all, I am struggling with this. These tools. Okay. No. Okay. All right, there we go. Smell. I normally don't have to use these tools, but my pen's not working for some reason. The gustatory picks up taste. Here we go. And vestibular cortex, these are going to pick up, so obviously we've done touch, sight, sound, smell, and taste. We've done our 5 senses. And I'm not telling you we have a 6th sense, but we do have this sort of area that is also heavily involved in balance and equilibrium. It's in the ear. And so the vestibular cortex really helps you with balance, just being able to maintain your balance. So like people that have vertigo, a lot of times it is an inner ear issue, and it has to do with the fluid in the ear. We'll talk way more about that in chapter 15 when we talk about the senses, okay? All right, so we're going to go through our sensory areas. We're going to start with this primary somatosensory cortex. And so that is this one right here, okay? So we say that this one sits just behind the central sulcus. So we say it's in the post-central gyrus. So remember when we talked about this primary motor cortex, we called it the pre-central gyrus. So now we're in the post-central gyrus. It's after that central sulcus. This one's going to get information from the skin, touch information from your muscles, and this one is also going to exhibit spatial discrimination. So what region of the body has been stimulated? So this picture here, so we'll come back to this one. So remember we talked about how in this primary motor cortex over here on the left side in red, we could see how this homunculus was kind of drawn out there and what areas of the body was involved in motor movement. Well, we also have a homunculus for this somatosensory cortex as well. So now we're going to look at the one on the right, which is in blue. So this is sensory information. So this is picking up touch sensation from different areas of the body. So again, we've been able to map out what parts of that cortex pick up that information from different areas of the body. Like if somebody touches your hand, what part of the cortex is picking that up? If somebody touches your face, or if you bite your tongue, what part of the cortex picks that up? And so you can see here that, again, this is still the same thing. We call it a homunculus. And again, it's out of order. You know, you can see there's the intestines and the tongue and the teeth, and then there's a face. So it is definitely out of order. And again, you're also going to notice that some areas are larger than others, the way they're drawn on here. Like you can see the whole like leg. Here's the leg and here's the torso. Those are really small, and look how huge the hand is, and the foot is really big. So in this case, when we're talking about sensation, the larger it is, the more sensation you have there. right? So like your hands, you have lots of nerve endings in your hands. Same thing with your feet. I went with my girlfriends a couple weeks ago and we had pedicures and I'd laugh through the whole thing. I can't. It just is so ticklish. Um, the tongue, you bite your tongue, it hurts so bad. You have so many nerve endings there. And same thing, like the teeth and the face, tons of nerve endings. So the larger it is, the more sensation you have, the more you're picking that up. Some of the other sensory areas that we have sort of towards the back of the brain. So again, we have this somatosensory association area. So remember, for all these areas, there's going to be an association area helping you interpret. So typically, because association areas are storing memories, they're going to be very close to the primary area. So we have this primary somatosensory cortex right here, and the association area is right next to it. Okay, you're going to see the same thing in the visual areas. You'll see the same thing in taste. So all the association areas are right next to them, storing those memories. So then what kind of memories can you store about touch? You can store memories related to size, texture, relationship of parts. And my favorite example of this is my cell phone in my purse. So I'm not a big purse person. I have a friend that just spent $2,000 on a purse. I would never do that. In fact, I just had to buy a new one because mine broke. The strap broke when I walked into Target. And so I was like, well, I'm in Target. I'll get a new one. I'm not a purse person. I don't change my purse out all the time. I have one. When it breaks or looks bad, I get a new one. But I always have really, really good intentions when I buy a purse. Like I want something very functional with lots of pockets, and I always don't get that. It's really hard to find a purse with lots of pockets that's functional. So inevitably I get a purse that's like this big giant bag. It's like a black hole and all my stuff just goes in it. And so a lot of times I'll be out and my phone will ring and my phone's in my purse with my wallet and my keys and the baby wipes and 1,000 other things. And I'll do that thing where I like unzip my purse and I shove my hand in and I don't even look and I just fish around in there for my phone. And I know when I have my phone, right? I know what it feels like. I know the relationship of parts. I know the texture. I know what it feels like in my hand without even seeing it. That is the somatosensory association cortex. I know what that feels like. I have restored memories of that. Same thing with my keys. If I fish my hand down into my bag, I know when I've got my keys in my hand. I know what those feel like, right? So it helps you form this understanding of that touch stimulus. So that is the association area. So some other areas that we have, some other sensory areas, obviously it's our other 4 senses, right? So we did touch. We also have our primary visual cortex. This is going to receive visual information from the retinas. So this typically is in the way back, it's in the occipital lobe of the cortex. Primary visual area is picking up the information from the retina. The visual association area helps you interpret what you're seeing. So it helps you determine the color and the form, and if there's movement. It helps you remember what that face looks like, right? Like I know what my son's face looks like. I know what my husband's face looks like. And that's from the visual association area. So where that's located is going to be way back here. So here's our primary visual cortex, picking up information about sight from the retina. And then this right here around it, that's the association area. So remember, they're always really close to each other. We also have auditory areas picking up sound. So we have a primary auditory cortex. This is going to receive information from the cochlea, which is in your ear, related to pitch and loudness, location. And then the association area stores memories of sounds. So it helps you to perceive that sound. I joke with my mom all the time. My mom can't hear. She has hearing aids now, but I joke with her that she must not have her auditory association area isn't working. because we'll be out and like somebody's cell phone will ring and they'll have like a custom ringer on their phone, like a different ringtone, which my mom doesn't even know how to change her ringtone. So she has like the generic iPhone ringtone. And someone's phone will ring and it'll be playing like music. And my mom will start looking for her phone in her purse. And I'm like, Mom, that is not your phone. You know that's not your phone. So she cannot remember what her phone sounds like. So I feel like she might have a problem in her auditory association area. So our other areas, so we've done touch, we've done sight, we've done sound. We also have smell, we have our olfactory cortex. Again, it also has an association area. We have taste, we have this gustatory cortex and its association area. And we also have a visceral sensory area, which helps us perceive our organs. You know, are we hungry? Are we full? And then we have a vestibular cortex, which is involved in equilibrium or I like to say balance. Okay, so those are all of the different areas that we have. Now, one thing about our cortex is that we do have this lateralization. I mentioned this earlier at the start of lecture. There's always a division of labor among your hemispheres, meaning one hemisphere is always going to be dominant. So you have a right and a left hemisphere, but typically one hemisphere is dominant and does the majority of the work. So usually it's the hemisphere that's dominant for language. Now, if you remember, Broca's area, your speech area, is only in the left hemisphere. So typically, the left hemisphere controls things like language, math, logic. 90% of people are what we call left-brained, meaning they are dominant in their left hemisphere. Now, remember, I also told you that your brain acts contralaterally. So the left hemisphere controls the right side of your body. So for most people, if you are left-brained, if left hemisphere dominant, you're going to be right-handed, right? Because it's contralateral. So typically, people that are right-handed are left-brained. If you are right hemisphere dominant, the right hemisphere controls things like creativity and emotions, right? This is why artists a lot of times are very emotional. Only about 10% of people truly are right-brained. But again, if you're right-brained, a lot of times you're left-handed. Now, this is not always the case, right? If you're left-handed, that doesn't necessarily mean that you're right-brained, but it is one way that we can tell. Now, there is something called bilateral dominance. People who are bilaterally dominant a lot of times are ambidextrous, meaning they can write with the right and with their left hand. And usually it looks really good either way, right? So I know some of you are like, well, I broke my arm in third grade. I broke my right arm in third grade and I had to learn how to write with my left hand so I can kind of do it. That's not the same thing, right? If you oftentimes switch your pen from hand to hand as you're writing, that's ambidextrous. The problem, we know in anatomy, right? Too much of anything is always bad, right? So it is really important that one hemisphere is dominant because otherwise, people who have bilateral dominance, those two hemispheres are fighting over simple tasks. So, for example, reading, you're trying to read, it's a simple task. And normally the left hemisphere would take over and do that. But the right hemisphere is fighting for control and it results in learning disabilities. So, a lot of times people that are bilaterally dominant have disabilities like dyslexia. It causes letters to flip around. It's because those hemispheres are fighting over control. So I'm going to go back over into our whiteboard and we're going to just add on to this, right? So we just talked about the cerebrum, okay? And I told you that we have our cortex, right? And remember the cortex is gray matter. And we talked about in the cortex, we have motor areas. We talked about how we have sensory areas and how our sensory areas, we also have these association areas. Okay, so we've just gone through our cortex. That's all we've talked about so far. And all the different areas in the cortex. Again, they're all gray matter, so they don't have a lot of myelin within those axons. Their job is not to send signals really fast. They're more interpreting the information that you're getting or sending out. So now we're going to move down in our cerebrum, and we're going to move down from the gray matter. Just under that, you have white matter. Remember, what makes white matter white is all of the myelinated axons. So you have a lot of myelinated axons here in the cerebral white matter. And the job of these is communication. So when I say that word communication, I don't necessarily mean you communicating. I mean neurons communicating with each other, sending signals very quickly between different areas of the brain and even between the brain and the spinal cord. So sending signals back and forth between areas of the brain or up and down between the brain and spinal cord. That's why you have a lot of myelin there, sending signals quickly. So a lot of myelinated fibers and tracts. So remember, we know these terms, right? Fibers and tracts, these are basically groups of axons together. And we classify all of these based on the direction that the fibers are running. So in the brain, in this cerebral white matter, we have commissural fibers, association fibers, and projection fibers. Again, these all have a name based on where they're sending the signals. So let's go through each of these. Remember that term fiber just is a group of axons, myelinated axons running together in the brain and spinal cord. Okay, so let's walk through our commissural fibers. So commissural fibers, this is areas of white matter that run back and forth between the two hemispheres, between the right and left hemisphere. So truly, this is how your right and left hemisphere can communicate with each other. They're connecting the gray matter of the cortex from the right and left hemisphere to each other, okay? And so a good example of this is something called the corpus callosum, which you're going to look at in lab this week. You're going to have to identify this in lab. So the corpus callosum is this big chunk of white matter that just sends signals back and forth. Okay, so let me show you a picture of this. Let me clear this drawing off here so you can see better. So in this case, let's look here at our commissural fibers. So the commissural fibers are in green. Okay, so on the left, you can see an artist's drawing, and on the right, this is a preserved specimen. So the commissural fibers, what you're looking at is a brain where like the front half of the brain has been cut off and removed. Okay. And so in green, you can see here, this is our white matter. These are our commissural fibers, and they're running back and forth between the right and the left hemisphere. Now, if we look at the image on the right, you can see the same thing, right? These are our commissural fibers. And notice that it's all white matter right there. It's very light in color. Okay. And that one specifically is called the corpus callosum. It's connecting the right and left hemisphere together. And you can see it's connecting these areas of gray matter in the cortex that we just talked about, okay, between the right and left hemisphere. Now, if we look at the brain from a sagittal cut, right, if we cut it down the middle and look at it, we can see these commissural fibers here. And this is how you're going to see it in lab. You're going to see it from this sort of sagittal view, from a lateral view. And so you can see right here, this is the corpus callosum. It's the same thing. The fibers are running back and forth. This big area of white matter. Okay, so that's commissural fibers. They connect right and left hemispheres. Association fibers connect parts of the same hemisphere. Okay, so let's look at those. We'll look at these over here. So association fibers are the ones that are in red, or this pink color, you can see here. So you can see here are the association fibers, and you can see that they are truly just connecting different areas of gray matter within the same hemisphere. Okay. So those are association fibers. I like to think of these when I see the word association. So remember when we just talked about association areas in the gray matter, they help you interpret. So that should make sense, right? You have these fibers that are sending the information really quickly from the association area to that particular sensory area and back. Okay, so association fibers. And you can also see them here, right, in the preserved specimen. If we look at it from the sagittal view, you can see them here. Okay, again, within the same hemisphere, just connecting these different areas of gray matter. And then lastly, our projection fibers. Projection fibers are going to connect the cortex to everything else, basically from the cortex all the way down to the spinal cord. Okay, so in this one, they're in purple, or sort of this blue color. So our projection fibers are these guys right here. So you can see here, and they're coming all the way down. So they're connecting areas of the cortex all the way down into that spinal cord. Okay, so that's a projection fiber. So it's running from the cortex down to the spinal cord. And if you look down here at the bottom, you can see them really well here as well, right? So they're running all the way from the cortex down into that spinal cord. Now, this area right in here, you can see where they're sort of radiating out from. We call that, and I have this on the previous slide as well, the corona radiata, which literally means the radiating crown. So it truly looks like it's radiating out from this one point. Okay. So again, this is our white matter. And the white matter is there to send signals either from one hemisphere to the other, within the same hemisphere, or down into the spinal cord. It is sending signals very quickly. Okay, so we have our association fibers, our commissural fibers, and our projection fibers. So, whoops, I didn't mean to do that. Let's go back to our whiteboard and let's add these in here. Okay, so we have cortex. We also have our white matter just underneath. And again, our white matter is made up of these fiber tracks. So again, we have our commissural fibers, projection fibers, and association fibers. Okay, so those are our different fiber tracks in our white matter. And then the last thing we're going to talk about still in the cerebrum is an area called the basal nuclei. These are deep pockets of gray matter. Okay, so let's jump back to our PowerPoint and talk about that. All right, so our basal nuclei are these deep pockets of gray matter, and they're found in both the right and the left hemisphere. And these are really important because they help us perform several activities at once. So it allows us to sort of multitask. It helps with movements. It helps us start and stop movements. It helps us monitor the intensity of our movements so they're not too wild or jerky or crazy. Disorders of the basal nuclei is essentially how we determined what the basal nuclei do. So disorders like Huntington's or Parkinson's disease, both of these diseases affect the basal nuclei. And in both of these disorders, we see wild, jerky movement, sort of uncontrolled movements, makes it very hard to perform activities. And so both of these are affecting this area of the brain. So that should make sense, right? If they're destroying this area of the brain, it's going to cause you not to be able to start and stop and monitor the intensity of an activity. Okay. So where do we find the basal nuclei? So, you know, we just walked through We've got our gray matter on the outside, right? That's our cortex. We've got this white matter just underneath, sending signals very quickly. And the basal nuclei are these deep pockets of gray matter. So like here and here. Now in this, they're colored pink here. But you can see these are very deep inside the brain. You can also see them here. So you can see these deep pockets of gray matter. Okay. So now we're going to move away from the cortex and we're going to move down and talk a little bit about this area called the diencephalon. So the diencephalon is, if we're looking at this image, it's everything that is this purple color in this image. So it is this area. Nope, sorry. It is this area right in here. Okay. So it is composed of the thalamus. Okay. And the thalamus, let me clear this drawing out. The thalamus is this right here is the thalamus. The hypothalamus, hypo means below. Here is the hypothalamus. And the epithalamus, epi means above or on top. The epithalamus is back here. Okay, so we have a thalamus, a hypothalamus, and an epithalamus. So let's walk through those. So we're going to start with the thalamus. The thalamus is actually the biggest part of the diencephalon, makes up about 80% of the diencephalon. It can sort information, edit information as it is coming from the body to the cortex. So remember we just looked at this corona radiata. Let me close out of here and I'll scroll back here. So remember down in here, I'm just going to use my pointer. So down in here, remember this corona radiata. So we are sending this information all the way out to our cortex. I want you to look at where it's radiating out from. It is radiating out from this little ball looking structure right here. That is the thalamus. So the thalamus, and this is how I always remember it. So again, oops, here we go. The thalamus, here's that ball looking structure right here. I always think of the thalamus as sort of like the FedEx. Information is coming up from the spinal cord and the brainstem, and it goes to the thalamus. And the thalamus goes, this is information about sight. I'm going to send it back here. This is information about touch. I'm going to send it up here. This is information about sound. I'm going to send that over here. So it's like the FedEx in that it is getting all this information, then it's sorting it, it's editing it, and it's sending it where it needs to go. Okay, it's like the shipping center of your brain. So that is the job of the thalamus. I always think of the thalamus as sort of like the FedEx. So I'm going to write that in here just because I think it makes it a little easier to remember. It is like the FedEx for your brain. And so this one is mediating all the senses that are coming in, all the sensations, all your motor activities that are going out. It helps with cortical arousal. It also helps with learning and memory as well. Now the hypothalamus sits just under the thalamus. It's kind of under and in front of. And this one is really involved in homeostasis. So it is regulating all of those things that you have no control over that help you maintain homeostasis. So it's going to regulate things like your blood pressure, your heart rate, pupil size, vasoconstriction, vasodilation, digestive tract motility. All of these are things that you have no control over consciously, but they have to constantly be regulated to maintain homeostasis, to maintain your blood pressure, to maintain your heart rate, right? So your hypothalamus is very involved in homeostasis. It is also very involved in emotional response. So fear, rage, pleasure, that should make sense, right? Because what happens to your heart rate when you're scared? It goes up. What happens to your blood pressure when you're scared? it goes up, right? You go to the doctor, a lot of people have white coat syndrome, where when they go to the doctor, their blood pressure is elevated. It's because they're scared, right? So your emotions are intimately linked with a lot of these homeostatic mechanisms, these autonomic controls, because they play a huge role in those. So it makes sense that your hypothalamus is regulating not only all of these homeostatic controls, but also it plays a role in those emotions as well. Okay, those are definitely tied together. It regulates things like temperature, hunger, thirst, sleep-wake cycles. It also plays a really big role in your endocrine system. And we will talk about this in chapter 16. So your hypothalamus is the boss of the pituitary gland. Your hypothalamus is constantly monitoring your blood, looking at what's in your blood. And if there might be a hormone that's low in your blood or something that's low in your blood, your hypothalamus picks that up and tells the pituitary gland, hey, we're low on XYZ. You need to release it. We need to fix it. So the hypothalamus is like the boss of the pituitary gland. So these are playing a very big role in the endocrine system and releasing hormones, specifically hormones like the antidiuretic hormone and oxytocin. Now we also have the diencephalon, which is this little dude right here. So this is our diencephalon. It sits, the epithalamus. The whole thing is the diencephalon, the thalamus, the hypothalamus, and the epithalamus. But the epithalamus is this little guy that sits sort of behind and on top of the thalamus. I am going to turn off my video because it seems to be lagging. I think my internet's lagging a little bit. All right, so this epithalamus really helps to regulate the sleep-wake cycle. It really helps to tell the pineal gland when to release melatonin. So the pineal gland is this little pink structure. And so the epithalamus is sort of the boss of the pineal gland. It tells it when to secrete melatonin. And I'm sure you know what melatonin is, right? You can buy melatonin at the store. You can take melatonin gummies. And a lot of people take melatonin if they cannot get to sleep. And so you naturally release melatonin, and it is released by that pineal gland. And the boss of that is the epithalamus. So let's pop back to our whiteboard and fill that in. All right, so remember we have in our diencephalon, we have the thalamus, which is the big structure. Again, this is sort of like the FedEx. We have the hypothalamus. And the hypothalamus is involved, I'm going to put a star on it, homeostasis. Regulates all of those autonomic functions. And then we have an epithalamus, which regulates and controls the pineal gland, which helps with sleep-wake cycles. All right, so we've done the cerebrum, we've done the diencephalon. Now we're going to move into the brainstem, and we'll talk about the brainstem. So the brainstem has three parts to it. It has the midbrain, the pons, and the medulla oblongata. These make up about 2.5% of the total brain mass. And the brainstem, it is providing a pathway for all of these axons, these fiber tracks. So let me underline that. So it's providing a pathway for these axons, these fiber tracks to run between the brain and spinal cord. Okay. We are in the next chapter going to be talking about nerves, cranial nerves, nerves that come off the brain. You have 12 cranial nerves. 10 of them come from the brainstem. The other 2 come off just above the brainstem, but 10 of them are coming off the brainstem. So that means that your brainstem is very involved with innervating the head and the neck. So again, we have our midbrain, our pons, and our medulla oblongata. And so this image, it's a little bit weird what you're looking at down here. You can see that we have the whole brain here. And if we take off the entire cerebrum, you can see we're left here with the diencephalon in purple and the brainstem here in green, right? And so really all we're concerned about is the brainstem. We're just going to look at this green part. Okay, and so the brainstem has 3 parts. It has the sort of upper part right here, which is called the midbrain. It has this middle part right here called the pons, and the bottom part is the brainstem. Okay, sorry, the medulla oblongata. So all 3 of those are the brainstem. Okay, the midbrain, the pons, and the medulla oblongata. And so we're going to talk about these 3 parts. We're going to start at the top, right? That's our midbrain. So all we've done in this image is we've just turned this. And so again, we're looking right here. This is our midbrain. Okay, again, in purple, this is our diencephalon up here. This is actually the thalamus, that lump that acts like the FedEx. You can see that here. So if we're looking at our midbrain, this is the pons, this big hump, this is our second, and this is our medulla oblongata down here, that third part we're going to talk about. So the midbrain at the top, the midbrain actually looks different depending on whether we look at it from the front, like this view. This is an anterior or ventral view. If we look at it from the back, or we look deep inside. So the midbrain is a little more complicated than the pons or the medulla oblongata. All of these structures, regardless of which one we're looking at, the whole point of the brainstem, everything in green, is sending fiber tracks where they need to go, sending axons where they need to go. And so I think your book did a good job with this image in that you can kind of see, like here, if we look, you kind of see these little striations in here. They're actually trying to show you where those fiber tracks are running. So you can see here, if you look at the midbrain from the front, these fiber tracks, they're sending information up to the cortex, right? If you look at the pons, you can see these fiber tracks are running in a different way. So we're just going to look right here at the midbrain. Now in the front of the midbrain, these structures right here, we call these cerebral peduncles. The term ped means foot. So these literally look like little feet holding up the cerebrum. And their job is to send signals from the spinal cord all the way up to the cortex. Okay. If we were to turn this around and we look at the midbrain from the back, so this is it from the back right here, what we see is we see these little humps right here, and there's 4 of them. This is called, from the back, this is called the corpora quadragemina. So quad means 4, and gemina, think of sign, right? Hey baby, what's your sign? Gemini literally means twins, if anybody's a Gemini. So the quadragemina is 4 twins, the 4 twins. And it does, it looks like 4 little balls right in here. Okay, so the corpora quadragemina. This is heavily involved in coordinating head-eye movements. So like, for example, if you're sitting at, my son plays hockey, and if I'm sitting at the ice rink and the puck goes by, I'm going to follow it with my eyes. And once it gets out of my field of vision, I'm going to follow it then with my head so I can catch up with it. And so that's the corpora quadragemina. It is helping with head-eye movements. It coordinates those together. It also helps with the startle reflex. Now, if we look deep inside the midbrain, we have 2 other structures. One is called, let me see if I have a picture. Okay, one is called the substantia nigra. So the substantia nigra is right here. This one gets its name. It actually tells you the color of it right here. Nigra means black. So the substantia nigra is black or really dark in color. Okay. And the reason, let's pop back over here. The reason is because the substantia nigra has a lot of neurons in it that release dopamine. And a precursor for dopamine, in order to make dopamine, we have to have melanin. Y'all should remember what melanin is. Melanin is the same pigment, that dark pigment that we have in our skin that protects against UV light. So in order to make dopamine, you have to have melanin. And so this is why the substantia nigra has that dark appearance. It has a lot of melanin in it so that your neurons can make dopamine. Now we also have another area within this deep part of the midbrain called the red nuclei. And the red nuclei also are named for their color. So the red nuclei are right here. And the red nuclei are red in color because they have a lot of capillaries and they have a lot of blood flow. So it gives them this sort of red appearance. Okay. So now we're going to move down the brainstem, right? And we're going to look at this area just underneath the midbrain. This area is called the pons. And again, I think your book did a good job because it's trying to show you, see all these striations? It's trying to show you what way the pons is sending information. It is a relay center. Every part of your brainstem is a relay center, right? It's either sending information up to the cortex, or in this case, the pons, you can see it's not sending information up. It's sending information back. And I know this image is small, but you can see the pons right here. And behind the pons is this little guy right here. That is the cerebellum. So the pons is connecting the motor cortex to the cerebellum. And then lastly is that elongated big portion just underneath the pons called the medulla oblongata. The medulla oblongata also acts like a relay center, like all these other areas of the brainstem. So it is relaying information to the cerebellum, but it also regulates homeostasis. Okay, so the hypothalamus regulates homeostasis, and the medulla oblongata regulates homeostasis. So the medulla oblongata is doing a lot of the same things that the hypothalamus is doing. It is changing blood vessel diameter. It is adjusting the heart rate. It is controlling your breathing rate, vasoconstriction, vasodilation. It is also your vomiting, hiccuping, swallowing, coughing, sneezing center. And so it might seem weird to think that you have two areas of your brain controlling homeostasis, but you want two areas doing that. All of these homeostatic functions are vital for you to survive, right? Your heart rate, vasoconstriction, blood pressure, these are all so important. And so instead of having one area of your brain controlling that, you have two. So it is very similar to the hypothalamus. Because it's the relay center. So you really have two areas controlling these really vital functions. So let's go back to our whiteboard and we'll add this in to our brainstem. So again, we have in our brainstem, we have the midbrain. We have the pons. And we have the medulla. And the medulla oblongata also functions in homeostasis, just like over here, the hypothalamus does. Okay. Now, all of these areas of your brainstem, these are all relay centers. So that's their job, is to send signals back and forth. Now, obviously, we talked about a lot of the different areas in the midbrain, right? The cerebral peduncles, the corpora quadrigemini, the red nuclei, and the substantia nigra. So we did talk about a lot of different areas in the midbrain. All right, so let's pop back to our PowerPoint and we'll finish out our brain and we'll talk about the cerebellum. All right, so the cerebellum makes up about 11% of the mass of your brain. This is that structure sort of in the back, just under the cerebrum. And the cerebellum functions in timing of skeletal muscle contractions so that you get nice, smooth, coordinated movements. So basically what your cerebellum is doing is it is constantly looking at your body and knowing the position of your body. And it is sending that position like a blueprint up to your cortex. And it's telling your cortex, this is the position of our body right now. So every second it is telling your cortex, this is the position of our body. So that when your cortex wants to move and wants to send information about movement, it can send the appropriate response based on the current position of your body. So your cerebellum is really important in helping you get these nice, smooth, coordinated movements. So let's pop back to our whiteboard and we'll just fill that in. Okay, so I'm just going to give you the function of the cerebellum. So again, this is just smooth, coordinated movements. So, obviously this is not a full outline that we did here, but I do think this is really helpful just so that you can see what areas of the brain are part of each of these 4 big structures that we walked through. Okay. All right, so we're just going to walk through for the rest of the lecture this morning, we're just going to walk through the protection of your brain and just a few disorders. So in terms of protection, remember I told you your nervous tissue is really, really soft. It's almost like jello. So it's protected by bones. It is protected by these meninges. These are coverings on top of the brain and spinal cord. It is protected by fluid. And also we have a blood-brain barrier. So we're going to go through each of those. I'm not going to spend a lot of time on bones because we've already done bones back in chapter 7. You have cranial bones that surround your brain. You have temporal, parietal, occipital, frontal, sphenoid, and ethmoid bones that completely surround the brain. And your spinal cord, you've got 30 vertebrae surrounding your spinal cord. Remember, your spinal cord runs through that vertebral foramen. So there is nowhere where your nervous tissue is not protected by bone. Okay, it is completely protected. Now you also have these meninges, and the meninges sit essentially directly on top of your brain and spinal cord. And we have 3 meninges that sit on top of the brain and spinal cord. The outermost meninge is called the dura mater. And I remember that because I think durable. So this is the outermost layer. It is really tough. It's like leather. It's made of this fibrous connective tissue. Just under that, you have something called the arachnoid matter. And I remember this one because I think arachnid, spider. So this one is the middle layer, and it actually has these sort of little leg-like projections that stick down from it, and it almost looks like little spider legs. So that's how it gets its name, arachnoid matter. And so where those little legs sit down, we call that the subarachnoid space, right? That should make sense. Sub is under or below. So this is just the space below the arachnoid matter. It's just allowing those little legs, those little spider legs to sort of penetrate down. And it's filled with cerebral spinal fluid. And then the innermost layer is called the pia. Pia means literally loving mother. So this is the innermost meninge. It sits directly on top of the brain and spinal cord. It is a very thin, delicate connective tissue. And it actually sort of caresses the brain and spinal cord. It sits right on top. It dips down into all of those sulci, goes over all of the gyri. And so this is why we call this the pia mater, the loving mother, because it truly looks like it's sort of caressing the brain and spinal cord. So here are our meninges. This is definitely not the best picture. I have mentioned this to Pearson multiple times. I don't like this one, mostly because it's really hard to tell our three different layers apart. So what you're looking at on the outside, like you can see this is our skin. You can see the periosteum of the bone, and then here's our bone, right? This is our flat bone. You can see it looks like a sandwich right here. So just underneath the cranial bones, you have this layer here. It's this gray color, and that is the dura mater. So you can see the dura mater right here, okay? It's actually, the dura mater actually has these two layers to it. You don't need to know those two layers. You just need to know the dura mater. Okay, so it is this really, really thick layer in here, and it is the outermost meninge. Again, it's very tough, very durable. Just under that, I'm going to do it in blue, you have this layer called the arachnoid matter. And the arachnoid matter is the one that's sort of translucent right here. So you can see it here. Sits right under the dura mater. And again, arachnoid, arachnid, think spider. And I told you it's because it has these little spider leg projections. You can see them really well in the image here. And that space in there, that's called the subarachnoid space. It's filled with cerebral spinal fluid. And then the very innermost layer is this pia mater, loving mother, and it sits right on top of the brain and spinal cord. And you can see it's really thin and delicate. It kind of dips into all of those sulci. Okay. And so I do want to mention a disorder that does affect the meninges, meningitis. Itis is inflammation of, so anytime you have inflammation of the meninges, that's meningitis. This is diagnosed by looking at the cerebral spinal fluid for microbes. There is viral and bacterial meningitis, so there's two different kinds. This is a really big threat. And the reason is because, remember, your immune system cells cannot get into the brain because of the blood-brain barrier. And so this is a really big threat because if you have an infection in your meninges, it can very easily spread from the meninges into the nervous tissue where you do not have immune system cells, right? We have microglia, but we don't have immune system cells. We don't get those white blood cells in that area. And if it gets into your brain, it leads to a condition called encephalitis, which is inflammation of the brain. Now, if your brain starts to swell, it has nowhere to go because you have these cranial bones protecting it. So that can lead to death. It can lead to, definitely lead to deficits from the swelling. Now you do have this cerebral spinal fluid also as protection. So you have meninges, you have bones. Cerebral spinal fluid is actually made from blood filtrate, and it is made by a structure called the choroid plexus. And the choroid plexus just takes your blood, filters it, and creates the cerebral spinal fluid that can then circulate through all of the spaces in the brain. Remember, a ventricle is a space. It can also circulate through the central canal of the spinal cord, which is a space in there, and also in that subarachnoid space. So any hollow openings are going to be filled with cerebral spinal fluid because nervous tissue is really soft. It's like jello. So if you tried to have a space in there, the nervous tissue would just collapse into that space. So we have to fill it up with fluid. This, because it's found in the subarachnoid space, also gives this nice cushion to your brain and spinal cord. So surrounding the entire brain in that subarachnoid space, you have cerebral spinal fluid. So it's really going to help. It basically allows the brain to sort of float in the cranial cavity. It prevents the brain from crushing under its own weight. It also protects it from blows and trauma. Also, your cerebral spinal fluid can help to nourish the brain and send signals. help you send some chemical signals. One of the ways that I remember that the brain is sort of floating in the cranial cavity is that sometimes women, when they have a baby, when they go into labor, they'll have what's called an epidural. An epidural is just an anesthetic so that they don't feel the labor pains quite as bad. An epidural is supposed to go into the space above the dura mater, but sometimes they can puncture through that dura mater. And if that happens, most of the time your body will heal that puncture. You'll create a little blood clot and heal it and seal it very quickly. But sometimes you do not seal that puncture well, and you will leak cerebral spinal fluid out of that cranial cavity. It continues to leak out from the brain and spinal cord, and the brain will start to actually sag in the cranial cavity because you're losing fluid. Think of like pulling the plug out of your drain. You're losing fluid, and instead of your brain floating up there, it's now starting to sag, and it creates a terrible headache. We call this a post-dural puncture headache. A lot of times what they'll do for women in this case is they'll actually take out blood. So they'll take blood out of their bodies, and they'll do another long needle in the same space, and they'll try to do what's called a blood patch. They'll basically push the blood into that area, hoping that it'll clot over that hole. Eventually your body will heal and will seal that hole, but it might take time, in which case you just have a really, really bad headache and there's nothing they can do for you. All right, we have a few more things to walk through in terms of protection. One thing I do want to mention about cerebral spinal fluid coming from that choroid plexus. So your body is constantly making cerebral spinal fluid from the choroid plexus. It is made, and then it's also drained. It's made and drained at the same rate. So usually you don't have like an excess accumulation of cerebral spinal fluid, or you're not losing cerebral spinal fluid, right? You have a constant rate. There is a condition called hydrocephalus, and hydrocephalus literally means water, and cephalo is brain, literally means water on the brain. This is when the choroid plexus continues to make cerebral spinal fluid, but it is not being drained, and it causes it to start to accumulate in the cranial cavity. Now, babies that have hydrocephalus have an enlarged head. You can see the picture here, and that's actually great for them because the skull bones have not fused together yet. As the fluid accumulates, those skull bones just separate. And so there's very little deficits with babies with this condition. In fact, they can do surgery and put in a little shunt that allows the fluid to drain, and then they're fine. Now, the shunt usually will have to be replaced throughout their life, but they can be fine. Adults with hydrocephalus usually will have brain damage because the cranial bones have fused together. And once you start to get that pressure in there, there's nowhere for it to go, and it'll push on the brain and cause deficits. Now, the last form of protection I'll mention on your brain is the blood-brain barrier. This is a selective barrier. It allows some things to pass, but not everything. So that's why we say it is a selective barrier. Things that can go through are things like nutrients. Those can move through. Anything that can pass through a plasma membrane can pass through a blood-brain barrier. So lipids and nutrients, those can all pass through a plasma membrane, so those can pass through the blood-brain barrier. Anesthetics can pass through the blood-brain barrier. Things that can't pass through the blood-brain barrier would be things like wastes, right? Those can't pass through the blood-brain barrier. And this is truly formed by tight junctions in the capillaries. The capillaries have these really, really tight junctions holding them together. We say there's a continuous endothelium there, and it prevents things in your blood from making it through your bloodstream into your brain. Now, we do not have a blood-brain barrier in some areas of your brain that are controlling homeostasis. So in places like the hypothalamus, you're not going to have a blood-brain barrier. In places like the medulla oblongata, you're not going to have a blood-brain barrier. So I wrote the vomiting center and the hypothalamus. The vomiting center is the medulla oblongata. So those two that we starred that are involved in homeostasis, those do not have a blood-brain barrier. And that's because these areas need to constantly monitor the chemical composition of your blood so that they can adjust hormones or that they can induce things like vomiting if needed. All other places in your brain do have a blood-brain barrier. So the last thing that I'll mention is just a few disorders that are associated with the brain. These are some traumatic brain injuries. They are listed in here from least severe. And I say they're least severe. I mean, a concussion is still a big deal, right? These are still affecting your brain. To most severe, right? So least severe to most severe. So a concussion, the word here, temporary, is why this is the least severe of all of these. A concussion usually results from a blow to the head, and it causes a temporary alteration of brain function. So you might forget where you are. You might not wake up for a minute, and then you kind of wake up. Sometimes what comes with a concussion would be vomiting afterwards, so you have to be monitored pretty closely. A contusion. A contusion is also caused by a blow to the head, but this is more serious because, notice the word here, this results in permanent neurological damage. So you will have permanent deficits from a contusion. A contusion, typically when we hear the word contusion, we think of a bruise, right? You have bleeding in that area. So that's essentially what's happening here. You've broken blood vessels and you're bleeding, and it's resulting in a permanent neurological damage. Similar to a subdural hemorrhage. Now, a subdural hemorrhage, hemorrhaging is bleeding into the brain, and this would be a hemorrhage where you have broken blood vessels under the dura mater and you are truly bleeding into the brain. And then the most severe is cerebral edema. This is swelling of the brain from a head injury. And again, anytime your brain swells, it has nowhere to go. So you do have deficits from that. All right, so a few disorders that I'll mention are things like strokes. A stroke is also termed a cerebrovascular accident. We just call them strokes. And a stroke is caused by any time you block blood flow to the brain. So this could be because you have a blood clot in an area, or maybe the blood vessel is being compressed. Maybe there's a tumor that's pushing on blood vessels that are supplying areas of the brain. Or you have atherosclerosis, and you've got some plaque buildup and narrowing of the blood vessels that are feeding this nervous tissue. All of those things can lead to blocking blood flow. Now, the term blocking blood flow is called ischemia. Now, everyone has experienced ischemia before. If your leg has fallen asleep or your arm, that tingly sensation you get, it is always caused by blocking blood flow. So maybe you're like laying on your arm funny. I can't sit crisscross applesauce because my feet will fall asleep really quickly when I do that. So clearly when I do that, I am pinching a blood vessel that is supplying my feet. So we all have experienced ischemia at some point, and we get that numbness, tingly feeling. The problem is that when this happens in the brain, nervous tissue has a really high metabolic rate. It has to have oxygen and nutrients, and it has to have it all the time. So if you experience ischemia in the brain, you will have deficits. So it usually results in paralysis or deficits. You can have a partial recovery after a stroke, and that's because, let's say, for example, we have sort of the synapse here where we have a couple neurons in a chain, right? We have 1, 2, 3 neurons. And let's say someone has a stroke and this neuron right here dies. Well, if that neuron dies and it's gone, well, you're not going to be able to finish that synapse. But over time, with physical therapy, this neuron here, this first neuron, might be able to regenerate. That axon, it can grow longer and it can kind of fill in the gap. And so this is why we might see in people who've had a stroke, after they've gone through physical therapy, they can have a partial recovery. Okay. Now there is something called a transient ischemic attack. So I'm going to write that down. It's a mouthful. It, for short, is a TIA. Transient just means reversible. Ischemic, we're blocking blood flow. So these are a lot of times what we call mini strokes. So these are temporary episodes of reversible ischemia. These are red flags. If somebody is having transient ischemic attacks or mini strokes, there is a bigger stroke on their horizon. So they need to see a doctor. A lot of times a doctor will put that patient on blood thinners. Sometimes they'll even do like ultrasounds of their carotid arteries, making sure that those are open, that they don't have atherosclerosis and plaque buildup, that they're not blocking blood flow. If that's the case, if they do have atherosclerosis, a lot of times they can do surgery and they can clean that out. So the treatment for stroke, if someone has had a stroke, the approved treatment that we have right now is something called tissue plasminogen activator, call it TPA for short. This is a medication that dissolves blood clots in the brain. This is actually something that your own body makes as well, that helps you dissolve blood clots when you don't need them anymore. But if someone has had a stroke, if they are found and tissue plasminogen activator is administered within 3 hours of the onset of the stroke, it can dissolve the blood clot, and a lot of times that person will have very minimal deficits. Unfortunately, people that have strokes, a lot of times if it's a massive stroke or if they're not found for a while and they can't get help, if it's been, longer than 3 hours, TPA is not very effective. So it has to be administered relatively quickly. So obviously there's some limitations with this treatment. So under study right now, there are studies where they're using immature neurons and implanting those in the brain to help people that have deficits from strokes, or even coaxing adult brain stem cells to replace damaged neurons. So these are both under study right now because of the limitations of TPA. I'll also mention another brain disorder is Alzheimer's disease. Alzheimer's is a progressive condition that affects the brain and results in dementia. Someone who has dementia doesn't necessarily have Alzheimer's. So dementia is essentially a symptom of Alzheimer's disease. People can get dementia. It's not uncommon for elderly individuals when they have, for example, a urinary tract infection. They will exhibit signs of dementia. But once the urinary tract infection is cleared up, that symptom will go away. Right. So dementia is just a symptom of Alzheimer's disease. What Alzheimer's is caused from is protein plaques that lodge between neurons. So again, if we think about a synapse between our neurons, here's a neuron and here's one, we rely on this area right here, the synapse, the synaptic cleft, where our presynaptic neuron has to release neurotransmitters into the cleft, and the postsynaptic neuron has to be able to pick it up for the signal to continue, right? And so what happens in someone with Alzheimer's disease is they get these big protein plaques that lodge between these two neurons. And what happens is, even if this presynaptic neuron releases a neurotransmitter, this plaque is in the way. And so this postsynaptic neuron never really sees the neurotransmitter, doesn't really pick it up. And so this is why someone with Alzheimer's has dementia. It is because of these protein plaques making these synapses pretty much useless. Another brain disorder that we see is Parkinson's disease. Parkinson's disease is caused by the degeneration of dopamine releasing neurons. So you have some neurons that release dopamine specifically in places like the substantia nigra, right? It's in the midbrain. So we know it's in the midbrain, the substantia nigra. That's an N. And that's an I, sorry. The substantia nigra, remember it's black in color because you have to have melanin to make dopamine. And so someone with Parkinson's disease, you know, like Michael J. Fox has Parkinson's disease, and you'll notice he has like tremors. It's really hard for him to control some of his movements. And so in the substantia nigra, those neurons that release dopamine have degenerated. We don't know what causes this. We don't know what causes Parkinson's disease. It could be genetic, could be something that is turned on when you get a certain virus or an illness. We don't know. The treatments for this so far, something called levodopa or L-dopa. This is basically synthetic dopamine. So obviously, if the dopamine releasing neurons are degenerating, causing you to have these sort of tremors and those uncontrollable shakes, if we give a patient levodopa or synthetic dopamine, it can alleviate the symptoms, but that is only in the short term. So this is not a long-term treatment. It only works for a little while, especially during early onset of Parkinson's. There is promising treatments using electrical stimulation where they actually will do surgery and implant little electrodes in areas of the brain that can shut down abnormal brain activity. And then also gene therapy. Gene therapy is a super promising treatment for any genetic disorder or any disorder where they have identified a particular gene. In gene therapy, essentially what they do is if they know the malfunctioning gene, they can get the correct sequence of genetic material inserted into a virus, and then they infect you with that virus. And as the virus replicates, it puts this correct gene into your cells. This is incredibly controversial because obviously we're changing somebody's genetic makeup. But for people with genetic disorders, this is so promising. Now this one, because we don't know exactly what causes Parkinson's, they're not really getting rid of it, but what they're doing with the gene therapy here is they're actually inserting the genes into a virus that then insert those particular genes into your adult brain stem cells, and it causes those brain cells to secrete GABA. Remember, GABA is a neurotransmitter that's an inhibitor, and it can shut down some of that abnormal activity and sort of turn off some of those tremors that we see. And I think this is our last one, is Huntington's disease. Huntington's disease is a fatal hereditary disorder. It is caused by the accumulation of the Huntington protein. And it causes the basal nuclei, these deep pockets of gray matter that are in the cerebrum, to start to degenerate. And so again, remember, your basal nuclei are really important in stopping activities. So if those degenerate, you no longer have the ability to do that. So the symptoms of Huntington's disease are wild, jerky movements. People say that it looks like you're dancing to your own music. It is sort of these uncontrolled movements. The treatment for Huntington's disease, there really isn't one. It is a fatal disorder. Usually someone with Huntington's disease dies, usually does not have a very long lifespan, usually dies in their 20s. The treatments are right now using stem cells. This is a very promising treatment, so embryonic stem cells. Again, anytime we're talking about gene therapy or stem cells therapy, it's just these are controversial treatments, but can be so effective for people with these disorders. Okay. All right. And so on Wednesday, we've done essentially all of the brain. If we go back and look at that whiteboard, we've talked all about these different areas of the brain. We talked about protection of the brain, and we talked about some disorders of the brain.